Hello, IB Bio students. So today we start topic 8. And as you can see here, topic 8 is divided into metabolism, cellular respiration, and photosynthesis. So today we're going to delve into some areas of metabolism. And here you have the essential idea of 8.1, which is metabolic reactions are regulated in response to the cell's need. And then we have a few understandings, applications, and skills. Okay, so today we're going to learn about metabolic pathways, and we're also going to learn about activation energy. So we're going to focus on the first two understandings here. All right, let's get started. So the first understanding here is that metabolic pathways consist of chains and cycles of enzyme catalyzed reactions. So let's explore a little further as to what that means. So the first thing you need to know is the definition of metabolism. This is the sum of all reactions that occur within an organism in order to maintain life. So the metabolism in you is all the chemical reactions that are happening in your body. They could be chain reactions like glycolysis and cellular respiration, where glucose is converted into pyruvate, or it can be in the form of cycles, like the Krebs cycle in cellular respiration, or the Calvin cycle in photosynthesis. Okay? So for these reactions to be regulated, they need to be controlled by a specific enzyme. So we're also going to look at how these enzymes occur and how they function within a metabolic pathway. And then we're also going to see those intermediates that are in between the between the substrate and the reactant. All right, so here we see an example of a chain metabolic pathway. So here you see the intermediates, right? So here we have a reactant right here, which is A, and here we see some intermediates, B, C, and D. And sometimes the chain can end with a product, right? Or sometimes, like this could be the product, D. Or sometimes that end product can also be an intermediate that could go into a cycle. And we see this happening in cellular respiration. We see how pyruvate is actually an intermediate within glycolysis. And then it goes into the Krebs cycle like this, right, to be able to release the carbon dioxide that is released during the Krebs cycle. So these are just types of metabolic pathways. We're going to look at them in more detail in just a moment. Actually, not till 8.2, but we can start talking about them. All right, so activation energy. So you've probably seen this before. You've probably seen it in chemistry. If you took physics, you probably saw this as well. But what is activation energy, and what is the formula for activation energy? So the essential understanding is that enzymes lower the activation energy of the chemical reaction that they catalyze. So the activation energy, the formula is E subscript A. And you have to keep in mind that every chemical reaction requires a certain amount of energy in order to proceed. So think of that energy to proceed, that energy found within the bonds. Okay, so that's something important to remember as well. So what do enzymes do? Enzymes speed up the rate of chemical reaction by lowering the activation energy. So let me move this up a little bit, and you're able to see here, if the dry frogs were the reactants and the wet frogs were the product, and this barrier of rocks represents the activation energy, a normal reaction that is not enzymatically driven is going to have a high energy barrier. So it's going to take a lot of energy for that frog to jump over the barrier. So therefore the reaction could proceed, right? But if it does proceed, it's going to proceed at a slow rate, if at all, right? due to a prohibited activation energy threshold. So the energy is very big here for these reactants to break their bonds and turn into products. But if you have an enzyme catalyzed reaction, that enzyme is going to lower the activation energy, so that barrier for these frogs, which are reactants, to jump over. So the reaction is going to proceed at a significantly faster rate, right? Because you're lowering the activation energy threshold. So these reactants, which are the dry frogs, could be turned into products, which are the wet frogs. Okay, so that's pretty much how the activation energy works. Now, keep in mind that the enzyme binds to the substrate, right? So that's what's happening here. So in an enzyme catalyzed reaction, there is an enzyme that is going to bind to this reactant or substrate, and that is going to stress and destabilize the bonds in the substrate. So that's what actually lowers the energy barrier, right? That means that the substrate that is now disrupted in some form, that substrate is going to require less energy to be converted into a product, and therefore the reaction happens faster, right? So the energy barrier that gets lowered is the energy found within the bonds of the reactants, right? There's a destabilization of those energy bonds, and therefore the reactant can be turned into a product much faster. All right, so there are types of enzymatic reactions that you're responsible for understanding. So again, you've probably seen these before. So we have the exergonic reaction, and then we have the endergonic reaction. So these are two types of enzymatic reactions, or you can also call them two types of reaction pathways. So let's look at the exergonic reaction at first. So here you see that the reactants, they contain more energy than the products. So if this is the free energy, or the delta G, notice how the reactants are at a higher state here of free energy. So the reactants contain more energy than the products. Okay. Now, let's look at the reaction with no enzyme. So if the reaction or the reactants actually do break up without the help of the enzyme, notice that the activation energy, which is this hill, is going to be much higher, right? So the activation energy is right here without the enzyme. But if you do use an enzyme, then the activation energy is going to be lower, right? So now the hill is much lower, okay? So the dashed line represents how this reactant is going to break up with the help of an enzyme. Now, look at what's happening in this catabolic reaction. So it's catabolic because the reactants here are breaking down. And when they break down, what happens? Energy is released from the broken bonds. So this delta G here is positive because now energy is being released from the broken bonds of the reactants. Okay? So think of a catabolic reaction as taking one reactant there and breaking it up into two. Okay, so this would be an example of a catabolic reaction, or an exergonic reaction, or a positive delta G reaction. Now, in opposition here, we have an endergonic reaction, and an endergonic reaction is considered an anabolic reaction. That means that you actually have to build up or construct a product of some sort. So in an anabolic reaction, you are pretty much going the other way. So you're taking perhaps two reactants of some sort, right? And you're combining them into one product. So that's an example of an anabolic reaction. And look at what's happening here. The reactants contain less energy than the products. So notice how the reactants are much lower in the free energy scale here than the products are. Now, when these reactants are trying to build up that product, right? Well, what's happening, right? So you have a much higher energy of activation, of course, with no enzyme, a lower energy of activation with enzyme, right? But notice that energy is required nonetheless to synthesize the bonds between the molecules. okay? So then that means that you're going to require energy. You're going to require energy to build the bonds between these two molecules. So then that means that you have a negative delta G, or endergonic, right? So that's what that means. So endergonic reactions have a negative delta G. Exergonic reactions have a positive delta G. Exergonic reactions are considered catabolic, or breaking down, and energy is released from the broken bonds. And endergonic reactions are considered anabolic, or building up, and energy is required to synthesize the bonds within the molecules. Okay, so that's it for today.